Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. If you like this video, subscribe my channel and click on bell icon to get notification. In this video, I am going to explain one of the topic in cell cycle and cell division. It is cell cycle and its characters. Growth and reproduction. These are the two important characteristic feature of every cell and it is needed for all living organisms. Both multicellular and unicellular organisms, they can start their life with the help of a single cell. It is called zygote or fertilized egg cell. It is formed by the fusion of both male and female gametes during sexual reproduction. So during growth, the new cells are formed from a pre-existing cell with the help of cell division, and every newly produced cell again it undergoes next division. It is referred to as reproduction. All cells reproduced by dividing into two daughter cells from each parental cell. Then after completion of this division, it results in the formation of two daughter cells every time, and these two daughter cells can divide again and again. Before dividing into two daughter cells, so every cell it undergoes and it can perform different phases or different stages in cyclic form. The sequence of events by which a cell it can duplicate its genome in S phase and synthesis various constitute for cell, and eventually the cell can divide into two daughter cells in mitosis division. All these events can be observed in a cyclic pattern and it is referred to as cell cycle. And the cell cycle is considered as it is a period or a time in between formation of two daughter cells, and further the daughter cells can divide to continue the cyclic process. So all the processes it includes cell division, DNA replication and cell growth, and have to take place in coordinating way. This coordinating manner it helps to ensure correct division and the formation of progeny cells. These progeny cells it consists of intact genome. That means the parental cell which having the genome the same copy of a genome present even in daughter cells. When a cell enters into cell cycle, this cell it undergoes various phases, and in these phases the cell it preparing for cell division. The cell growth it is due to increasing the cytoplasmic content, and this process it continuously occur throughout the cell cycle. But the DNA synthesis it takes place only during a specific stage of cell cycle. During this DNA synthesis, the replicated chromosome it consists of DNA, and this replicated DNA it will be distributed into two daughter cells in its nuclei, and it undergoes a complex series of events during cell division. So all these events which are take place during cell cycle, these are under control of genes. Phases of the cell cycle. By taking a typical eukaryotic cell, the cell cycle it consists of two basic phases, and these are interphase and M phase. M phase is also referred to as mitotic phase. In interphase, the interphase it is defined. It is a period or phase in between two successive M phases. In this interphase, it deals with various changes. These changes can be seen inside of the cell, especially in both cytoplasm and nucleus before it enters into cell division. During this interphase, the cell prepare itself to perform next division. And this interphase is also referred to as biosynthetic phase because in this phase the cell it duplicates various cell organelles along with the DNA present inside the nucleus, so that it is referred to as biosynthetic phase, and this phase is the most active stage of cell cycle. In this interphase, actually the phase is very long and it is considered as a non-dividing phase, growth phase of cell cycle, and this phase it occupies nearly 95% of duration in cell cycle. In this interphase, due to having of various changes inside of the cell, outside we cannot observe any changes related to cell division, so that it is referred to as resting phase, because it's the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. During this interphase, it deals with the changes, and all these changes can be observed in cell and inside the nucleus before it enters into next cell division. This interphase further it classified into three stages. In this, the first stage is G1 phase, second one is S phase, third phase is G2 phase. G1 phase is also called as gap 1 phase. S phase is also called as synthesis phase, and G2 phase is also called as gap 2 phase. These phases are seen in interphase. In case of G1 phase or gap 1 phase, in G1 phase, the correspondence it is present in between mitosis and initiation of DNA replication. So during cell cycle, after completion of mitosis division, immediately G1 phase is started and it ends due to initiation of DNA replication in S phase. During G1 phase, the cell it is metabolically more active. At the same time, the cell it continuously grow in size, but there is no replication or synthesis of DNA in G1 phase. The cell grows in size continuously. Along with that, many changes take place in cytoplasm and in nucleoplasm of the cell by synthesizing nucleotides, ATP molecule, proteins, amino acids and RNA. And all these are the requirements for the synthesis of DNA in S phase. So along with that, most of the cell organelles are duplicated in this phase, and cell divides frequently. Then such type of cells which divide frequently, these cells have shorter G1 phase, but sometimes if cells do not undergo division frequently in such cells, the G1 phase is longer. Coming to second phase, it is S phase or synthesis phase. In this S phase or synthesis phase, it is marked due to initiation of DNA synthesis or DNA replication. So during this time, the DNA it will duplicated with the help of a process, it is called DNA replication. During this DNA replication, the DNA both strands acts as a template strands for the synthesis of new strand. At this time, the amount of DNA it will double. Initially, if the DNA it is denoted with the 2C, and after completion of S phase, now the amount of DNA it is denoted with 4C. That means the amount of DNA, it will be double. Before S phase, in G1 phase, the number of chromosomes are 4. But here, when you observe both these chromosomes, in that chromosomes amount of DNA, it will be double due to DNA replication. During this S phase, amount of DNA, it will be double, but there is no change in number of chromosomes, because in G1 phase, if it consists of 4 chromosomes, after S phase also it consists of a 4 chromosome itself. So in S phase, there is no change in chromosomal number. By taking one example, if we take a onion cell, it is diploid in condition with 16 number of chromosomes. The 16 number of chromosomes are seen in G1 phase, that is before S phase, and even after S phase also the same number of chromosomes 16 are maintained in diploid nucleus. So with this example, we can understand that there is no change in number of chromosomes, but the amount of DNA, it will be double. Along with this DNA replication, some of the histone proteins which are associated with DNA packaging, these are also synthesized in S phase itself. The newly formed DNA, it forms a new chromatin fiber, but it remains attached to its pair. In this diagram, it is one of the chromatin fiber before S phase, and it is also one more chromatin fiber of chromosome in S phase. So after completion of S phase, due to DNA replication, this chromatin fiber, it will be duplicated and it becomes into 2 chromatin fibers, but still both these are attached with centromere. And this chromatin material, it condenses to form 2 chromatids in a chromosome, and both these are called as sister chromatids. They can attach us with centromere. But in case of animal cells, during S phase itself, along with the DNA replication, so the DNA replication, it takes place in nucleus, but outside of the nucleus in cytoplasm, in case of animal cell, the centriole duplication takes place. So these are the main changes during S phase or synthesis phase in cell cycle. Along with that, the centrioles, these are the main cell organelles seen only in animal cells, and these are required for M phase. And during this M phase, these centrioles can form spindle fibers. But such type of centrioles are completely absent in plant cells. Coming to G2 phase or gap 2 phase. In this phase, it is considered as a second gap phase, and it is present in between S phase and M phase of cell cycle. In this phase, we can observe the growth of cell continuously, but when the G2 phase is initiated or started, immediately DNA replication process, it will be stopped. But in that cell, the synthesis of RNA and proteins, it will be continued, and these are required for next phase of cell cycle. So during the G2 phase, one of the important protein, it is called tubulin protein, and this tubulin protein, it takes place in G2 phase, and this tubulin proteins which are synthesized in G2 phase, it helps in division phase for the formation of spindle fibers. Along with that, during cell cycle, in special conditions, we can observe one of the special phase. It is called G0 phase or quiescent phase. Remember, this phase not commonly present in cell cycle. And some of the cells in adult animals, they do not appear to exhibit division like heart cells and nerve cells, so that heart cells and nerve cells are considered as undividing cells. They never divides further. Along with that, many another cells present in animals, they can occasionally perform cell division as needed to replace some of the lost cells in their body due to injuries or cell death. At this time, we can observe a special phase in cell cycle. It is called G0 phase or quiescent phase. And in this G0 phase, with a normal cell division, new cells are formed in M phase. But these newly formed cells, it just enters into G1 phase, but immediately this will exit from cell cycle and then it enters into one inactive stage. And this inactive stage is referred to as quiescent stage or G0 phase. So during this stage, the cell it behave normally that at this time it is metabolically more active, but there is no longer proliferate, that means formation of new cells, and it is called to be as G0 phase, and it depends on the requirements of organisms. M phase, it is the second main phase in cell cycle, and M phase is also referred to as mitotic phase, and this phase it actually cell divides into two daughter cells, and the duration of this M phase is very less, and approximately less than 5% of time duration is there for M phase, and in this M phase one cell it completely divides into two daughter cells. In mitosis is a process in this one parental cell it can divides into two identical daughter cells, and these cells are identical to each other as parent. In this interphase, the cell it duplicates various components, and all these components are equally distributed in two daughter cells at the time of M phase or mitotic division. This M phase it consists of two important phases in this. The first phase is karyokinesis. So karyokinesis means the parental cell have well organized nucleus, and this nucleus it will divide and becomes into two daughter nuclei is called as karyokinesis. And this karyokinesis it is followed by cytokinesis, and in this cytokinesis one cell it will gradually divides into two daughter cells is called as cytokinesis, is also referred to as cytoplasmic division. During M phase, reorganization and separation of all components occur in a cell. When we observe the mitosis divisions, especially in case of animal cells, so mitotic cell division are seen only in diploid cells. But in case of plant cell, the mitosis division can be seen in both haploid and diploid cells, especially the lower group of plants like algae, bryophyta. In their body, it consists of haploid cells and it is a gametophytic plant body structure. Such type of haploid cells also can perform mitosis division. The period of cell cycle, generally it is varies from one organism to another organism and also from one cell to another cell. By taking some example, so a yeast cell, it is a unicellular fungi. It need 90 minutes to complete its cell cycle. So that the duration of cell cycle in yeast is 90 minutes. A typical human cell, it can divides into two daughter cells for every 24 hours. That means in human cell, 24 hours time is needed to complete one complete cell cycle. So these are some example for duration related to cell cycle. Thanks for watching.